Welcome back, students, to Chapter 12, Part B. In this section of this chapter, we're going to be looking exclusively at ion channels and their properties. So something for you to remember straight away is that ion channels have two properties which are unique to them. They are ion selective. That means they only allow certain ions to pass through the interior gaps, and they have a doorway, a gate, that can be opened and closed to turn the ion channel on and off. So how do we classify ion channels as being on and off? Well, the terms closed and open can be applied at this point. So this gate here on the left is closed because this part of the protein is unable to allow the passage of its ions. So this will be one domain of this protein complex. Another domain on the same protein is responsible for selectivity. That means it filters which type of ions can get through, and we'll see that on the next slide. But these two properties, selectivity and the fact that they're gated, is unique to ion channels as opposed to just simple membrane holes between cells that you see in other situations. So looking deeper at these ion channels, in this particular case a potassium channel, we notice that the proteins are arranged in a donut shape to allow the formation of a central pore. And the pore has many functions along its length. The most critical location will be here, in this case, where we have the selectivity filter. This selectivity filter basically brings the amino acids which form the protein donut very close to the central pore. In fact, there's contact being made between some of the atoms of the amino acids making these proteins and the actual ion being transported. And as the ion goes through, it doesn't go through in a straight line as indicated here. It jiggles around, bouncing back and forth. And only if it fits into the crevices created by the atoms of the amino acids will it have a suitable path through to the other side. If other ions or entities inside the cell wish to sneak through this particular filter, they would have to negotiate not just the selectivity filter region, but also the vestibule. And the vestibule is an area which is responsible for removing water molecules which are surrounding the ion. This is called hydration. So the ion is only able to pass through once the water molecules are ripped off of its surface at this point here. And that's achieved again by these residues of the amino acids that form the lining of the selectivity filter. Once the ion has passed through to the other side, the water molecules are picked up afresh from that environment to rehydrate the molecule. If sodium tried to get through this gap, it would have the wrong dimensions and the wrong form of hydration to negotiate through these three different regions. The importance of charge to living cells cannot be overemphasized. The membrane potential, as the word suggests, potential, is a storage of energy that can be tapped into at any point thereafter. So one thing that we need to understand is that for the vast majority of the cytosol and for the vast majority of the external environment, the charges are balanced because positive charge and negative charge interact constantly to form a matrix of balanced charge. But whenever there's a membrane that has ion channels or pumps included in its structure, those regions can establish a very localized region of charge imbalance. So the positive charge that you see on the left-hand side in panel B is very close to the membrane. Should you travel a bit further, that potential will disappear and become negated. Likewise, on the other side of the membrane, we have negative charge. So to be more precise, a negative charge is normally found on the inside of the cell, and positive charge is normally found on the outside of a cell in its resting configuration. So this is a law of how biology works. It's very rare, if at all, to find positive charge on the inside of a cell in a resting cell. As we'll see later, it can happen during action potentials and other types of signaling, but in a resting cell, you find this configuration, with the outside of the cell being on the left and the inside of the cell, the cytosol, being on the right. Panel A of this slide shows us a single potassium channel of a membrane and the situation around that particular channel protein. If the channel protein is closed and there is no potential for charge to move from one side to the other, then there is zero membrane potential. Even though work is available to be utilized, there's no way for the molecules to pass through in order to go downhill and change that potential energy into some form of kinetic energy. However, when the channel opens due to some type of stimulus, there is a flow of charge represented by the movement of potassium ions indicated here in yellow. The reason we have two arrows is that in this case, for potassium, there are two forces at work. The first, the yellow arrow, is because there's much more potassium on the inside of the cell than out, and the potassium is forced to go down its concentration gradient. This is chemistry. The blue arrow is physics. This is the force of the extra total charge outside the cell being highly positive, and that's due to sodium and many other types of positive charge. And that opposes further positive charge from joining them. So the blue arrow and the yellow arrow are fighting each other. And whenever they reach a balance, an equilibrium, that's the point at which there's no net movement of potassium further along its ion channel, even though the ion channel may be open. So that's when we have a membrane potential that exactly balances the tendency for potassium to leave. That's an important concept that we need to learn. Using some experimental data, some scientists in the past have come to conclusions about how to quantify the membrane potential that's generated on two sides of a membrane due to the presence of any one ion. And the Nernst equation does exactly that. So the V stands for the membrane potential in volts, and there's a constant number which we don't have to worry about, thank God. And then if you multiply that by the log 10 of this ratio, the concentration outside the cell of that ion and the concentration inside the cell of that ion. And that solution will give you the voltage, the membrane voltage due to that ion alone, if that ion alone is responsible for maintaining a membrane potential. Figure 25 from your textbook illustrates some important principle about ion channels in general. It comes back to what we talked about with regard to doors and pathways. When something is open, it's not completely open. And when something is shut, it's not completely shut because of the random motion of molecules at the molecular level. And this is illustrated nicely by this diagram. Here we're studying a single ion channel using a patch clamp technique where we can monitor the membrane potential over time. And as you can see, for this particular channel, it opens 3 times during a 27 millisecond period. And for most of the time, it's closed. So for this particular one, we would say that this channel is closed because for the majority of the time, it has no membrane potential passing across it. When it comes to classifying ion-gated channels, we can look at the way that they are opened in response to stimuli. So some of them are voltage-gated. So the ion channel itself is able to detect the voltage across the membrane and then respond to that by opening or closing. Another type is the ligand-gated ion channels, and they respond by opening when they bind a ligand. Now the reason we have 2 panels here, B and C, is because some of them bind material extracellularly, and others bind material intracellularly. So we have the two alternatives, but they're both the same. And then the third one that we see here, labeled D, are the mechanically gated ion channels. And in the mechanically gated ion channels, the force of nature is a distortion mechanically of the membrane itself. And that is illustrated beautifully by the inner ear of a human being, where the hair cells have projections on their surface, and those projections can be rubbed one way or the other by sound waves that move the tectorial membrane by pulling it and pushing it at different frequencies along its length. And that generates within the cell a distortion that allows the passage of sodium into the cell, and that causes a membrane potential to be generated at the base of the cell that's then transferred along the auditory nerves to the brain to be perceived as sound. That's further illustrated by this example here of a mimosa plant where touching the leaf, either by human touch or by insect, will cause the leaf to curl up into this shape. And that's a combination of that's a combination of mechanically gated and then ligand gated ion channels. So students should read that example in the textbook. Moving on rapidly, the next thing the textbook concentrates on are action potentials, which is a very specialized case of ion channel regulation of the activity of cells. So a typical neuron looks like this. It can be different, completely different, but this is a textbook example. So here we have the cell body with the dendrites carrying information towards the cell body, and then one of the specialized structures is an axon, and the axon is designed to carry information away from the cell body, and in this case it just subdivides at the termini. That leads to many branches, and each branch can then be attached to a muscle or another neuron or to a gland, thereby transmitting information in that direction. As I said earlier, most of our research has been done on the squid, and the giant squid in particular, because its axons are huge in comparison to human axons. And as the blue pages in the book illustrate, we can put our electrodes deep inside those huge diameter neurons to measure the membrane potential. And by doing experiments like this, we can measure how different ion channels open and close at different points to propagate an action potential along the length of this axon. The same panels indicate that if we change the concentration of sodium outside, the action potential can be then altered by various degrees depending on the starting combination of ions. As we'll see in class during a skit, the complex interaction between at least 3 different types of ion channels is responsible for propagating action potentials along axons in animals. The most important one is the sodium ion channel, which has 3 configurations that it transverses through when activated. So the normal resting axon has an inactivated sodium ion channel, which is closed to sodium migration. Upon stimulation by a voltage difference in the membrane, the voltage-gated ion channels open, the sodium ones, and they allow sodium to pass through, as indicated here. And then they reach a state of inactivation, where they are prevented from responding any further, irrespective of the change in the membrane potential. And this time period lasts for a predefined period until they are then reset to the closed configuration, pending further disturbance in the membrane potential. And that leads to a wave-like propagation along the axon that leads eventually to transmission of information to the far reaches of the axon. Once the signal reaches the end, the signal has then to be converted to a chemical signal, and that's done by the activity of voltage-gated calcium channels, which open and release calcium into the cytosol, and that causes in a complex manner the fusion of these vesicles with the membrane of the cell. So that releases acetylcholine in this case, and that acetylcholine then impacts the sodium ion channels, which are located on the other side of the synaptic cleft, and then that activates the secondary cell. So this is a pretty sophisticated process which we must learn, and that's illustrated in the remaining few slides that you see here. So this is your voltage-gated calcium ion channel, and that's activated by an action potential that travels down the axon, as we saw earlier. And once they're opened, the calcium rushes in and it immediately fuses the synaptic vesicles, which contain the neurotransmitter, with their own membranes, therefore releasing that into the cleft. And that's picked up as a ligand that binds to these ligand-gated sodium channels, which will then allow sodium to enter the cell. So this process does not always have to be in that particular fashion. So there are alternatives available. So some neurons can activate their neighboring neurons, other neurons can delay activation by acting in an opposite manner, by increasing the distance between the zero membrane potential and the minus membrane potential. Focusing deeper on the acetylcholine receptor, we can see that it's made of 5 subunits, and 2 of the subunits bind the acetylcholine, causing a conformational change in their structure, and that then allows the channel gate to be opened. And the distance between the region here is only suitable for the entry of sodium, not any other ion like potassium. So there's your specificity. And then the final slide just shows you some examples of ion channels that are found in the various cell types and their locations and their functions. So in summary, this chapter is pretty straightforward. It just talks about ion channels, and that's all we need to know for now. Thank you.